Hi everyone, this is Elizabeth McCormick. I'm a genetic counselor and research coordinator here in the Mitochondrial Medicine Frontier Program at the Children's Hospital of Philadelphia. And I am just going to talk a little bit today about the different tools and resources that we use when we're really trying to make sense of the genetic information that our patients come to us with. So before we get really into the tools and resources though, it's important to just talk about the genetics of mitochondrial disease. So I don't want to spend too much time on this because I'm sure if you haven't heard it this morning, you'll be hearing about it this afternoon. But it is important to have a general understanding so you know what we're talking about when we're reviewing the different tools that we'll be going over today. So we all know that primary mitochondrial disease can be caused by pathogenic or disease-causing variants in mitochondrial DNA or nuclear DNA. So if we start with mitochondrial DNA, as we have here, we know that mitochondrial DNA contains 37 genes, so it's much smaller than the nuclear DNA that has more than 20,000 genes. But the genes of the mitochondrial DNA are very important. So 13 of these 37 genes actually encode subunits of the electron transport chain enzyme complexes that are very important for generating energy. 2 genes encode rRNAs and 22 genes encode tRNAs. And these classes of genes are really important to really turn that genetic sequence in those 13 protein coding genes into those protein subunits. We also know that mitochondrial DNA is maternally inherited. There are no introns, so we don't have to worry about splicing as we do for nuclear DNA. And we also know that in mitochondrial DNA are the presence of haplogroups. So haplogroups are sets of single nucleotide variants or small indels that cluster together to form these fixed groups or haplogroups. And they have ancient origins. They're actually associated with particular lineages as humans migrated around the globe. So by definition, these variants are not pathogenic or not causing someone's medical concerns, but it could be that these variants arise independently of the haplogroup that they're classically associated with, perhaps in the setting of a different haplogroup. And in that case, it would be important to analyze that variant really as you otherwise would. And then we also know that primary mitochondrial disease can be caused by pathogenic variants in nuclear DNA. So similar to those 13 protein coding genes in mitochondrial DNA, nuclear DNA has genes that encode for other subunits of those electron transport chain enzyme complexes. Nuclear DNA also has genes with many other really important roles for the mitochondria to function properly. Several classes we have listed here. And there can really follow any inheritance pattern for nuclear DNA. So autosomal recessive, autosomal dominant, or X-linked. So even though we know that primary mitochondrial disease can be caused by pathogenic variants in nuclear DNA or mitochondrial DNA, the 1 or 2 variants that's causing someone's medical concerns is not always clear to us. We actually all have thousands and thousands of variants across both genomes, and that doesn't mean we all have thousands and thousands of different diseases. Most of those variants are benign, but the 1 or 2 that are driving or are causing someone's medical problems are not always clear to us. And that's where the resources that we'll be talking about today can really be helpful, really to just kind of gather evidence for or against variants causing medical concerns. So when we think about these resources, I want you to think about 2 specific scenarios. So the first is that you're a clinician following a person you think has primary mitochondrial disease. You send genetic testing to a clinical diagnostic laboratory, and you get a report that shows a change or a variant in a gene. It could be that that variant is what we call known pathogenic, has been seen in people before, and is known to be causing medical concerns. Or it could be that that variant is what we call of uncertain significance. So it's a change in a gene and it's there. We're just not sure if it's causing someone's medical problems or not. And there's a couple different ways that the tools can help you gather more information about those variants. So, for example, the tools can actually point you to case reports that have shown other people with this variant. So even if a variant, is known pathogenic, you might want to know what other people with that variant, what their medical concerns were, what you need to be on the lookout for your patient that you're seeing. Or if it's a variant of uncertain significance, you might want to gather more information about it. Two important pieces of information would be, the first is that what's the frequency in the general population. That can actually be a very powerful tool because we know mitochondrial disease is rare. So if your patient has a variant, but that variant is seen in 25% of the general population, well, that can't be causing all of your patients' medical concerns, because then 25% of the population should be having the same medical concerns. And we know mitochondrial disease isn't that common. So population data can definitely be helpful. And then these tools also have what we call in silico predictors, or really detailed computer algorithms that take several aspects of the variant into account and pull that information together to give you the best guess of the effects of that variant, if it's disease-causing or benign. These are not perfect by any means. They're really just to be used as a guide, but they can certainly be helpful as you're gathering evidence, again, for or against these variants being the cause of someone's medical concern. And then the second scenario you can think about is, again, you're a clinician that's seeing a patient that you suspect has a primary mitochondrial disease, and you send genetic testing and it comes back normal. That does not mean that your patient does not have a primary mitochondrial disease, because it's important to keep in mind that the clinical diagnostic labs are really good at reporting out variants in what we call known disease genes, or genes that have been shown to cause medical concerns when pathogenic variants are present in them. But clinical diagnostic labs, for the most part, can't report out, what we would call novel, or genes that haven't been recognized to cause medical concerns in people before. And who's to say that your patient has to have a cause that has been seen before? Maybe it's something that has yet to be recognized. So we'll go through some tools in that case that can help you analyze your patient's sequence data to see if we can find the answer. So this is just a table that I won't go into too much detail. This is a table from a paper that's actually in press at the current moment. And this is for mitochondrial DNA variants. This paper is from a project that our group and several other colleagues have been working on to standardize mitochondrial DNA variant classification and interpretation. But I think this table is useful when you think about resources for primary mitochondrial disease, and again, mitochondrial DNA, which this table focuses on. So the tools on the far left we have listed. These are the ones that we have found to be the most helpful. And then our first column is sequence data. So which of these tools have general population sequence data that you can get a sense of what the variant frequency is in the general population? The second column is, do these tools have those variant pathogenicity, or those in silico predictor tools that can give you, a really good guess of if that variant is disease-causing or not? And then the last column is, does the tool allow for sequence analysis? So not an individual variant, but someone's entire mitochondrial DNA sequence. And these tools can do such things as determine the haplogroup of that patient, or call out the variants in that sequence. So again, hopefully this will be useful to you if you're thinking about mitochondrial DNA variants in the future. And just to briefly run through a few of these tools, so MitoMap has really been the gold standard mitochondrial DNA variant tool and database over the last several decades. And that is led by Marie Lott and Dr. Doug Wallace and Dr. Vincent Procaccio, who work on that as well. And it's really a great resource of mitochondrial DNA variants as well as sequence data. MSeqDR is a tool led by Dr. Marnie Falk and Dr. Xiaolu Gai, with the bioinformatician behind the scenes is Dr. Li Shuang Shen. MitoSNP score is a tool out of the group from India, and then HMTDB is a tool developed by Dr. Adamanelli in Italy. So now if we're going back to our scenario number 1, where we have a mitochondrial DNA variant that's been reported in our patient, where can we go to gather more information about the variant? So I have here 3 example variants. The first is a known pathogenic variant, has been seen in many people that have primary mitochondrial disease. The second is a variant of uncertain significance, and the third is a variant. Then the tool we're going to use to learn more about these variants is MSeqDR. So the website is just MSeqDR.org, and when you go here, you will log in, as I have highlighted here in the right corner. And once you log in, there's many tools that you can play around with and that are useful, but for the purpose of learning more about these particular variants, we're going to start with MVTool. So what I did here was I copied and pasted the variants over, and now I'm going to click and annotate. So you'll see once you click that, you have a lot of information that's given to you, but I'm just going to draw your attention to the first box of information, and you'll see that the tool MSeqDR is able to pull information from MitoMap. So on the top line, this is our known pathogenic variant. You'll see that it's actually not present in those sequences in the MitoMap, or that MitoMap has access to. So that is always a clue that maybe this is a pathogenic variant, because again, it hasn't been seen in healthy people so far. Our second variant is seen in one individual. It's always a little bit tricky to interpret, because was this individual healthy and the variant truly is benign, or was this individual 20 years old when their sequence was put into this database and they didn't develop features until they were 40? So that can be a caveat with the population data as well. And then our third variant you'll see is actually present in more than 1,000 people. So that's fairly convincing that this variant is benign, because we know it would be a little bit far-fetched to believe that 1,000 people had the same primary mitochondrial disease as seen in your patient. And then you'll also see for our first variant, we get the MitoMap curations, or the pathogenicity assertion. So this variant has been associated with Lee syndrome and MELAS, and it has reached the confirmed category. And then this is the same screen as before. I just scrolled over a little bit, and you'll see it has HMTDB disease assertions and associations, as well as the mitotype predictors. Mitotype is a great tool to predict the effects of tRNA variants. So that's just, again, a quick overview of what MSeqDR can show you. I just wanted to show you the MitoMap resource real quick. So on this page, you will go to search. I typed in here our pathogenic variant, and you'll see the conditions that it's been associated with, that it hasn't been seen in the general population databases. And then if you were to click the references, you actually get a great listing of publications that have reported cases with this variant. So it's not exhaustive by any means, but it's definitely a great point. And now to move on to our second scenario, where you're a clinician and you have a patient with a strong suspicion of primary mitochondrial disease, but haven't been able to find the answer for that person to date. But say perhaps you have their raw exome data or their raw genome data that you want to analyze, or you have their mitochondrial DNA sequence data. How can you try and find the answer in those? So MSeqDR can help us with this as well. This time we're going to go down to the QuickMy tool. And here what you would do, we have detailed instructions on the screen. You would just upload the VCF files that contain the exome and perhaps the mitochondrial DNA sequence data. You can also put in your patient's medical concerns, and then when the program is analyzing the data, they'll actually give you back out genes or variants in genes that may have some overlap with the medical concerns seen in your patient. Always helpful. And then I just wanted to draw your attention to some other tools that we have that, again, if you have a mitochondrial DNA sequence, can help you call out the variants in that data as well as determine the group. And then just to finish up, I wanted to make sure everyone knew about this brand new resource that our group and others have worked on over the past year. And really what we did in this mitochondrial disease genes compendium with the editor, Dr. Marnie Falk, is several of us went through and really just put together a summary of many primary mitochondrial disease genes. So we included information from common features seen in those with pathogenic variants in this genes, to common variants in the genes, to what animal models exist for variants in this gene. So we're hoping it'll be a very kind of practical resource for those of you seeing individuals that have primary mitochondrial disease. So hopefully by now you find mitochondrial DNA variants and genomic variants in general a little less daunting now that you know how to just go about gathering more information on those variants and just try to make the most sense of the genetic information that we have on our patients with primary mitochondrial disease. And I would be happy to take any questions. Thank you.